So with Chapter 8, we're going to be looking at photosynthesis. In this world, we have different types of feeders. We have our autotrophs. So our autotrophs are going to be our self-feeders. Auto meaning self, troph meaning feeder. And then you have your heterotrophs. And these guys consume from others. And so the heterotrophs rely on our autotrophs to obtain their nutrients. And in particular with photosynthesis, we're going to be looking at photoautotrophs, which obtain their energy from light, photo, to make the food that they make. And our photoautotrophs tend to undergo photosynthesis. And so they make carbohydrates. Plants, cyanobacteria, remember our cyanobacteria are thought to be the precursors to our chloroplast, which is the organelle where photosynthesis takes place. Protists such as algae, unicellular euglena, all can carry out photosynthesis. And so there's an animation in your study area that you can go look at that shows the flow of carbon in producers, which are our photoautotrophs, consumers, which are our heterotrophs, and then our decomposers, which are our bacteria and fungi. The basic makeup of our leaf is this waxy cuticle on either side of the leaf. Within the waxy cuticle, we have these stomata, which are the openings into and out of the leaf. And so the stomata are made up of two guard cells, and the guard cells can open and close to conserve water. They would close, but then they can open to allow gas exchange, bringing CO2 into the leaf, letting the waste product of photosynthesis, oxygen, out of the leaf. Within the leaf, we have our mesophyll cells, and within the mesophyll cells, these darker green dots are our chloroplasts. Within the chloroplast, just like you saw with the mitochondria, you have an inner and outer membrane. You have this intermembrane space between the inner and outer membrane. You have these green pancake-like structures within the chloroplast called the thylakoid. And within each thylakoid, they have an internal space called the thylakoid space. Stacks of thylakoid are called the granum. Here you can see a photomicrograph of a mesophyll cells with all the chlorophyll in it. And here you can see a chloroplast with the thylakoids, these little flat stacks. Each little flat disc pancake is a thylakoid, and a stack of them is a granum. Just as we saw with cellular respiration, photosynthesis is a redox process. And so one of our reactants becomes reduced, meaning it gains electrons. When you gain electrons, you become more negative. Your overall charge is reduced. So you are reduced. So carbon dioxide is reduced to become glucose. Water is oxidized to become oxygen. And here you can see where all of the individual atoms go to form our new molecules. So the C, the carbon and the oxygen from carbon dioxide go into glucose and into water. And when the water is split, these hydrogens go to form the glucose, a molecule of water. But the oxygen that is released from water is the oxygen that is released from our photosynthetic organisms. And that is the oxygen that we rely on, that we breathe, and that we also rely on to carry out cellular respiration. So as was pointed out to you with your cellular respiration, It's the exact opposite of photosynthesis pretty much. Cellular respiration is taking glucose and oxygen, making water and CO2 and energy in the form of ATP. The energy that we are putting into our photosynthesis reaction is a photon of light. So photons are how you measure light energy. And so sunlight in particular fuels photosynthesis, and so a photon of light adds the energy. CO2 and water are then transformed into the glucose and oxygen that then goes into cellular respiration to make water, CO2, and ATP is the form of energy made by cellular respiration. Photosynthesis has two sets of reactions. With cellular respiration, we saw the three with glycolysis, the Krebs cycle, and then the electron transport chain. Photosynthesis starts with our light reactions, and these are going to take place in the membrane of the thylakoids. So these are our thylakoids, and you have the photosystems that the light reactions depend on embedded in the thylakoid membranes. So you have your reactants of a photon of light, water, NADP, ADP, and they all come into the light reactions, and the product that is released from the light reactions is oxygen. You also make ATP and NADPH. These are going to go into the next step of photosynthesis, the Calvin cycle, along with CO2. So ATP, NADPH, made in the light reactions, and NADPH is the same thing as the NAD, or similar, as an electron carrier that you saw back with cellular respiration. And so ATP, NADPH, a reducing agent, and then carbon dioxide, our carbon donor, all come into the Calvin cycle. It will need to turn 6 times to produce 1 molecule of sugar. And so here is a summary. We have a 2-stage reaction for photosynthesis. We have our light reactions, and then we have our Calvin cycle, which is our light independent reactions. With the light reactions, water is split as an electron donor and also provides the hydrogens, the protons, to run our ATP synthase, because you're going to see with photosynthesis, just like with cellular respiration, that you have a proton pump that is going to build a hydrogen gradient, and that hydrogen gradient, fueled by our electrons, is going to run back through the enzyme ATP synthase to make ATP. The oxygen is released as a product of our light reactions. Again, our electron acceptor is our NADP, so it is our electron carrier. It taxis our electrons to our second reaction, the Calvin cycle. And then the ATP is generated via chemiosmosis in our light reactions. And then that ATP and NADPH that were made from light energy, so we're converting this light energy into chemical energy in the form of ATP and NADPH, are then going to move into the second stage of photosynthesis, the Calvin cycle, along with CO2 to make our sugars. And so here is a visualization of that process. And so here is our sunlight striking our leaf. And within our leaf is our chloroplast. This is the thylakoids. This is our phospholipid bilayer here. And so you can see the phospholipid bilayer. We have our photosystem 2 and photosystem 1 embedded within the thylakoid membrane. These reactions make ATP and NADPH that are carried to the Calvin cycle. Water comes in, it is our electron donor, it splits, oxygen is released. So water is split, donates its electrons, oxygen's released. Those electrons run through photosystem 2 to make ATP, then run through photosystem 1 to make NADPH. Electrons donated, run through our electron chain, make ATP, NADPH, come to our Calvin cycle, and again we have to run the Calvin cycle 6 times. So this is our 6th cycle, and we'll make our 1 molecule of glucose from turning the Calvin cycle 6 times. ADP and NADP are recycled back to our thylakoid, our light-dependent reactions, to make more ATP and NADPH, coming back over into our Calvin cycle. So the photons of light that are used in photosynthesis come from a very narrow spectrum in our electromagnetic spectrum. So our electromagnetic spectrum includes gamma, X-ray, UV, infrared, microwaves, radio waves. But what photosynthesis can use is the visible light spectrum. Visible light spectrum runs from 380 to 750. And here you can see that 380 to 750 we have pigments. Pigments are the molecules in our thylakoids that are able to absorb the light and get excited. And so they can absorb the light and take that energy in. And so our main pigment is chlorophyll A. And then we have a second pigment, chlorophyll B. And then our accessory pigments are called our carotenoids. And these are the ones that you actually see in the fall. So in the fall, when light starts to shorten, the days start to change, the chlorophyll A and B start to die off, and then the carotenoids are able to see. So the carotenoids are what give us our fall leaf colors. But our main pigment and is in our reaction center is our chlorophyll A, and then chlorophyll B, and then our accessory pigments are the carotenoids. And you can see that chlorophyll A absorbs a lot of light here at 420, around 420, and again over here at around 680, 700 it absorbs. Same thing with chlorophyll B right here, probably around 460, and then again over here somewhere around 650, and then all the carotenoids are down here in these wavelengths. Notice that there is no light absorption from about 520 to 620. And so when light strikes the chlorophyll, all those areas that pigments are active and absorbing light are absorbed, but that area of the green yellow is reflected, and that's why we see plants as green, because they reflect the green yellow light and do not absorb in that spectrum. And so that's why we see plants as green. And so here we can see the efficiency of photosynthesis. So with this white light, you're at 100% efficiency. So you've got 100% efficiency right here. Now let's change the wavelengths. 400 to 430 goes to 91% efficiency. Then when you go to 430 to 460, 91% efficiency. And then when you go to the next wavelength, 460 to 490, drop to 83. Then when you go 490 to 520, drop to 62% efficiency. And then when you hit 520 to 550, 0% efficiency. And then when you move to the next wavelength, 550 to 580, still 0%. Next wavelength, 580, 610, still 0%. So from 520 to 610, 0% efficiency at those wavelengths in that visible light spectrum. When we move up to 610 to 640, get to 57% efficiency. Move up to 640 to 760, 670, 87. And then move from 670 to 700, 62% efficiency. And then again, that white light back here at the beginning, when you've got all of it on, 100% efficiency. So changing the wavelengths and where they absorb affect the efficiency of photosynthesis if you only had one wavelength at a time, or one group of wavelengths at a time. And we'll stop there and continue on in our next talk.